Hey, hi everyone. My name is Nicholas Prager. I'm the CEO of SmartCella. I'll be giving first a short background to the company and then spend most of the time in this talk around our delivery technology called the extraducer. And I'll just finalize with a few words around how we would partner with interested parties. So we're a global biotech working on targeted therapies by combining delivery solutions and advanced therapy development. We have two separate delivery technologies. One is an endovascular delivery device that I'll be talking mostly about today called the extraducer. And the other technology that we're working on as well is the cell-based delivery of mRNA. I will not be spending time on that today. We're also developing therapies utilizing the cell-based delivery technology is one leg of our pipeline of therapeutics. The other one is around more traditional stem cell therapies where we have heart cells as the most advanced project that's out-licensed to AstraZeneca since 2018 and ready to go into the clinic now. And we're also working on dopaminergic cells and looking at other platforms there. But again, I will not be speaking to those today. I'll be focusing on the extraducer. The company was founded in 2014. We're about 60 to 70 employees based in Stockholm. And we have raised so far a little in excess of 50 million euros. So again, it's a combination really of first-in-class delivery platforms with novel mRNA therapies and more traditional cell-based therapies that we believe is now really sort of at a very interesting intersection when most of us, I think, agree and start to feel that it's pretty well proven that the cell and gene therapies that all of us here are working on are very effective treatments. But it is a challenge still with delivery, and we believe that we have some of the tools that could really be interesting in that aspect. So the extraducer is what I'll be focusing on, and we're looking a lot at solid tumor indications. As you know, there's been a strong focus so far on rare diseases and blood-based cancers, but obviously, there is a hope that all these new therapies will also be able to target solid tumors in a wider way than it is today. But then we have this problem of surgery being difficult to fully remove the tumors, and it requires still secondary treatment in most cases. Chemotherapy with all the systemic side effects that come with it, and the problem of getting enough concentrations into the tumor. And radiation therapy, some of the same problems with very wide side effect issues. And then for the cell and gene therapy specifically, we have these problems of hostile tumor microenvironments that limit the effect of CAR-Ts and others, heterogeneous antigen expressions that mean that you're not really hitting necessarily all the cancer cells that you need to hit, and off-target and off-tumor toxicities. Most of these drugs are today administered through IV, traditional IV. That means non-specific, obviously, very much systemic exposure, insufficient dosage a lot of the time, particularly in the target organ, in the target tissue, and then coming with off-target toxicity. And we see with cell therapies that we get accumulation in the lungs, and with gene therapies that we get accumulation in the liver. And for mRNA, the problem is many times degradation before you get enough protein expression. So what we're suggesting instead is to look at this new technology where you use an endovascular technique to go in through the groin or the arm or the neck, and with standard equipment, guide catheters, navigate to where you want to get in the body directly to the tissue, an organ, or directly to a tumor, and then penetrate with this extremely sharp needle directly through the vessel wall, entering into the target tissue. Here we're talking about a tumor then, for example. To get very, very precise delivery, you can have exact dosing with multiple injections around the site, around the tumor. And And we can do this with a lot of different modalities. And this product is already FDA approved in its first version, and the second version is now about to be submitted for approval in the third quarter. And we have a lot of animal data, both in small and large animals. So it comes from the Karolinska Institute and a professor called Staffan Holmin, who had been working on the Seldinger technique, which is the traditional sort of catheter technique where you put in stents and all these things that have been done for several decades now. He was thinking about the idea that if you can penetrate the vessel from the outside, why wouldn't you be able to do it from the inside? And he came up with this extremely thin needle that's utilizing then standard guide catheters to get to where you want, and then inside the guide catheter forward this needle, which is about 1.8 meters long, thin as a hair, and hollow, which means that when you then go through the vascular wall with the needle, you have a micro work-in channel all the way from the outside where you can then inject now any payload that you want directly into the tissue. And this is the first time someone has done this. And it's the hole that is left when you withdraw the needle is so thin that blood can't pass under normal physiological pressure. That obviously means there's extremely limited, if any, risk of bleeding or thrombosis. And this is equipment that's being used that is available at any sort of Western hospital today in an angio lab. It requires extremely little teaching for people to be able to use this. We've done that over Teams in 10 minutes, and people are off running because this is sort of standard equipment that they're used to. As I mentioned, it's already FDA approved with 510(k), and version 2 is about to be filed. We're also filing, and that's for abdominal organs, and we're also filing an IDE in the third quarter this year for heart, for cardiac indications, and we have the technical documentation. The data packet is ready for any, basically, combination and device, with drug and device. And here's just a timeline on this. I haven't mentioned the brain. We've done animal studies on the brain, and it's looking super interesting, and you can all imagine the utility of being able to access the brain from the inside rather than having to go through the skull. We are leaving that a little bit behind on the development timeline, though, because of all the sensitivities with the brain. First in human trials will be conducted in September, is the plan to start this year. It's both our own study, where we're using the extraducer to deliver chemotherapy. So a generic drug that has a lot of problems with systemic side effects today in inoperable pancreatic tumors in that case. We're injecting then with the extraducer directly into the pancreas. That can obviously be done with any cancer drug that's targeting the pancreas. Instead, we're just doing it with a generic to prove the concept. So that's a phase 1/2 trial that will be starting in September. We're also starting human trials, a phase 2b trial in September with the first partner that we signed a global commercial deal with on a heart indication for regenerative medicine. So we can, as I said, then access difficult and risky to reach tissues and organs. That means solid tumors is an obvious prime target for us. It passes through the vascular wall, which means that we get direct access to tissue. It's very precise with conventional navigation and imaging methods, and there's no additional capex for the hospital to start using this immediately. It's very minimally invasive. The patient stays away through the whole procedure. Typically, it takes about 2 minutes to navigate to wherever you want in the body with these guide catheters, and then we can do 10 or 20 different injections during 30 to 40 minutes. So the patient will be in and out in less than an hour, and that's obviously really great news for fragile patients. It also makes multiple injections possible. As I said, it's both multiple injections in one procedure, but it also allows for repeat access, that you can come back and do repeat injections. So in our pancreatic cancer study, for example, we're going to do 4 different cycles with 2 weeks between them. Easy to use, I mentioned already, with minimal training. And we think that this can truly revolutionize how cell and gene therapy treatments are being delivered, and really with low barriers for the people that are actually performing these procedures. We're, as I said, modality agnostic, so we're looking at all kinds of therapies, both old and new ones. And for the cancer indications that we believe are most interesting and that we're focusing the partnering efforts around are in pancreas and kidney, heart, some more regeneration obviously, but liver and lungs. We're also looking at ovaries, and I mentioned the brain already. And the small molecules we're testing ourselves then in the pancreas, as I mentioned, and we have a regenerative medicine partner that's focusing on the heart, but we are interested in additional partnering discussions now. And if you think about them, where would it be suitable? Well, the earlier you are, obviously, it's very easy to plan. So in the preclinical stage, you can think about the off-target difficulties that you may be challenged with, or to overcome tumor microenvironments, and utilize this as a way to overcome those challenges. But we're also, like with the partner that we have already, they were coming in at the phase 2b stage because they were seeing that they could get even better expression by having such precise and direct delivery into the heart. And obviously for the heart, the alternative is also open heart surgery, and this is a completely different way of doing it, which is much more friendly, obviously, to the patient in doing it. We also recommend in looking at assets that for different reasons have been shelved because of off-target toxicity or not enough efficacy, because of inability to really get to the target tissue in enough concentrations. Patent line extension is another way of looking at it for mature drugs. Again, I mentioned the generic gemcitabine that we're doing the pancreatic tumor study with. That's an example of that. So there are different ways of looking at how you can partner with us with drugs at different stages of development. So how do we do this? Well, we work with non-exclusive licenses with upfronts, milestones, and royalties. We can support with filing data and with GLP tox studies. We will give priority on catheter supply to the early adopter partners, and the first licensing deal, as I said, has already been signed. So we're just getting out there now to start talking to a lot of different partners. We believe, as I said, that a lot of the new modalities, CAR-T, NK cells, or viral therapies, and the whole basically cell and gene therapy space is highly attractive for us as partners. The way we do it would be to look together with you at if there's a fit between your product and the extraducer. You would work with our business development team. We will then look at the technical fit, the timelines, and the freedom to operate, and then make sure that we can do proof of concept studies in different animal models. We have an incredible facility at the Karolinska Hospital that we have full access to through the inventor of this device, which is probably one of the best facilities in the world. There are all kinds of imaging and equipment available for analysis that you would like in an animal model. And then we'll talk about the filing support that we can provide, and after that then commence with commercial supply agreements and licensing agreements. My colleague Nina Bauer, our CBO, is here, and you're welcome to reach out to us if you would want to discuss potential partnerships around the extraducer. Thank you very much.